introduce the series, and then Claire's going to introduce you, and then you can get going, but you'll have to share the screen. Okay, I think so, that was recording. So welcome to this webinar series on microglia. I'm Guy Brown from the University of Cambridge, and my co-host today is Claire Butler. And the series consists of monthly talks of about 45 minutes by European scientists talking about their research on microglia, followed by a Q&A session. And today's talk is by Amanda Sierra from the Basque Centre for Neuroscience in Spain, and she's going to be talking about how microglial phagocytosis maintains tissue homeostasis. So during the talk, if you want to ask a question in the Q&A session at the end, just write the question in the chat function during the talk so you don't forget it, and address it to all of us. And then we'll deal with those questions at the end. Please keep your microphones muted and cameras turned off in order to preserve bandwidth. So can you turn your microphones and cameras off now? Okay, so I'm going to hand over now to Claire, who's just going to briefly introduce the speaker.
Yep. I'm so sorry if I completely butcher some of the Spanish in this, but I'm going to try. So Amanda graduated in biology at the Complutense University in Madrid, Spain, and then she obtained her PhD in neuroscience in 2003 at the Cajal Institute, also in Madrid. She moved to the USA for her postdoctoral training at the Rockefeller University in Stony Brook University in New York, and then at Baylor College of Medicine in Houston. In 2011, she is a research professor of the Basque Foundation and the head of the glial cell biology lab at the
Achucarro.
Close, almost got it. Center for Neuroscience in the Basque Country, Spain. Her research is focused on understanding the role of microglial phagocytosis in health and disease.
Thank you very much, Claire. And thank you, Brown, for the kind invitation. I think this series is really wonderful, and it's a great opportunity for all of us to meet and see each other. So I'm gonna start sharing my screen. I guess you can see my screen now. Is that okay? Yes. So I just want to start by briefly mentioning that we are almost 100 years above the 100-year anniversary of Microglia, which was last year, and this is the wonderful cover that my colleague Rosa Palvicelli created for this article that we published in Intrinsic Neuroscience. So for those of you who don't know, Achucarro was a very interesting person. He was the first Basque neuroscientist, and he was the mentor of Pío del Río Ortega, who was the person who in 1919, as I was saying, discovered microglia. So this is a very interesting story because there was a big feud at the personal and the scientific level between Río Ortega and Cajal, and we translated the original papers a few years ago with Helmut Kettermann. So I really encourage you to read this paper of how microglia was born. So microglia, as I was saying, I think the field has evolved tremendously in the last 10 or 15 years, and it's because we have really understood how much they interact with every other element of the brain parenchyma. They contact neural stem cells, participate in neuronal function, monitor and prune synapses, are also related to myelination, vasculogenesis, the blood-brain barrier, and of course they have the more traditional immune functions like inflammation and phagocytosis. And my lab is particularly interested in this process, how this engulfment of apoptotic cells occurs. And this is a cartoon that represents, this is very important to understand because this cartoon represents the different stages, because phagocytosis is really a process that occurs in time. So first, the apoptotic cell has to emit some signals that attract microglia. So these are called the find me signals. Microglial processes have to get there, then they have to form this pouch surrounding the apoptotic cell, and finally they have to engulf it and digest it. And my questions are really related to what happens next. What happens after microglia degrade an apoptotic cell? And I'm mostly interested in how this contributes to tissue homeostasis. And this is just a cartoon of showing how we usually visualize apoptotic cells and microglia. So you will see many of these images throughout my talk. You will see the nuclei in labeled with DAPI. You see the dots of euchromatin and the lax heterochromatin, and these are normal live neurons. And then you have apoptotic neurons that have a condensed or pycnotic nuclei. We can also see them with caspase-3, and you see that microglia usually extend these processes and engulf them, very sort of like a vulture will extend its neck and engulf the corpse. So how do we study phagocytosis? I'm going to take a step back for a second, and to study phagocytosis in the adult brain, what we use is the adult neurogenic cascade as a model. And why the adult neurogenic cascade? Because it's a place, the neurogenic niche in the adult hippocampus, where here in the subgranular zone, we have the neuroprogenitors that continuously proliferate and give rise to mature neurons. Throughout this process, which is sort of depicted here, and you can see the mother cells, the daughter cells, and here the mature granular neurons. Throughout this process, many of these neurons die. And when they die, they are engulfed by microglia. So again, this is how we will visualize it. This is the subgranular zone. Here we have all the live neurons, an apoptotic neuron sitting by this neural progenitor. It's one of these daughter cells because it's expressed in PSN-CAN, and you can see that it's surrounded by this 3D pouch of microglia that really surrounds the cell. So one of the basic quantifications that we use to determine how efficient microglia is what we call the phagocytic index, which is the proportion of apoptotic cells engulfed. And regardless of which method you use to determine this phagocytosis, you can see that the vast majority of apoptotic cells are already engaged in this process. So you see cells that are pycnotic in these orthogonal projections of confocal cystax. You can see that they are caspase, and you see this wide 3D pouch surrounding the cells. And in cases where it seems to be like later stages, you see usually smaller nuclei, a more dim ring of caspase, and a tighter pouch of microglia surrounding the cells. So this is what we found when I was a postdoc back in the US many years ago, is that the vast majority of these newborn cells undergo apoptosis, and they are immediately recognized and engulfed by these so-called resting or surveillant or unchallenged microglia. They don't need to be pre-activated in order to become efficient phagocytes. So I'm going to tell you a few stories, some published and some unpublished, about what we have learned over the years regarding this phagocytosis. So one of the things I always wondered, is what happens next? Is this a dead end? Microglia are just like the trash can, they just remove the apoptotic cells and that's the end of the story? Or what is actually the response of microglia once they eat all these apoptotic cells? So in order to understand this or try to answer this question, we've done different types of experiments, both combining in vitro and in vivo experiments. So I'm going to show you something that we published last year, and it's related to how phagocytosis impacts on the surrounding neurons. So to understand this, we first started, this was work by Irune Díaz-Aparicio during her PhD. We first started with an in vitro model of phagocytosis. We have primary cultures of microglia, and we feed them with apoptotic cells. And then at different time points, we can assess, yes, they are phagocytic and so on and so forth. And what we did, we did some gene array expression analysis. And from all that data that it's already published, I'm going to only show you that we could already see groups of genes when we did the gene ontology analysis of these genes, groups of genes that were related to neurogenesis. So this suggested that there was some sort of a feedback. So microglia would engulf the apoptotic neuron cells, and then they would somehow feed back into the neurogenesis. And we identified many of these potential genes that were regulators of neurogenesis that were released by phagocytic microglia. So it's not just one gene that is regulated, but actually we have peptides, hormones, trophic factors, cytokines, and quite a bunch. So it seems like a coordinated transcriptional program that is initiated in the phagocytic microglia. So like I was saying, I'm going to present you sort of quickly two experiments that we did, one in vitro and one in vivo. In vitro, what we did is we cultured the neural progenitors, the neurospheres, and we grow them with control media, with the media from control microglia or with the media from phagocytic microglia. And then we assessed what type of phenotype had these neural progenitors. And what we could see is the medium from the phagocytic microglia really turned a completely different lineage, let's say, in in the neuroprogenitors, and it turned them into these bipolar cells that were really high expressing nestin and GFAP. And for the most of the experiments that we could do, these are really related to the astrocytic lineage. So it's not the phagocytic media is really turning on neurogenesis instead of, astrogenesis instead of neurogenesis. Like I said, we did many experiments. One of the final experiments that we did is use this conditioned media and inject it directly into the hippocampus and see what would happen to the neurogenic cascade. So we injected this media in osmotic mini pumps for 6 days, and then we treated the animals with BRDU. BRDU is this analog of thymidine. So when you give it IP, it will incorporate into the DNA of the dividing cells. And if you then follow those cells for 4 weeks, you can assess their identity and see the neuroprogenitors, whether they have transformed into neurons or not. So as you could see, we have fewer BRDU cells, fewer neuron cells. And when we analyze them into categories, we could see fewer neuroblasts labeled with double cortin, and fewer BRDU cells that express NEUN, so fewer neurons. So the conditioned media of microglia, of the early phagocytic microglia, really turns off neurogenesis. To further understand how this process would work, we did another completely independent set of experiments. This was done by Iñaki Paris during his PhD. And we used different knockouts for phagocytosis receptors. So we've used MERTK and AXL in collaboration with Greg Lemke from La Jolla, and P2Y12 knockout mice with Beata Sperlak in Budapest. So I'm going to show you 3 strategies that we used. So first, we use an acute blockade. So we blocked specifically MERTK in microglia, and then we gave BRDU and waited only for 1 day. So this is an acute blockade and then an early assessment. And you can see that it has a strong impact on phagocytosis. We use another model in which we waited a little bit longer. It's exactly the same procedure, but then we waited 4 weeks and used, so it's an acute blockade, but later stage. And you can see again a blockade in phagocytosis. And then we have a chronic blockade because these are non-conditional knockouts, just constitutive knockouts for both double knockout MERTK XL and P2Y12 knockout. And again, in both cases, you could see a reduction in phagocytosis, although not as strong as we had with the acute models. So what happens to neurogenesis in these models? What happens is something very interesting, and it's a biphasic response. In the acute blockade, in the early phase, we have sort of what we expected based on the in vitro experiments. If we block phagocytosis, now we have more BRDU cells, right? Because the conditioned media from phagocytic microglia is anti-neurogenic. So we got what we expected, an increase in the number of BRDU cells. But if we waited for longer, and I hope you can really see here, now this increase at 1 day is not really translated by an increase at 4 days. And actually we have even fewer cells. So if you calculate the ratio from the cells you have here to the cells that have survived here, and from here to here, you can see that actually blocking phagocytosis is detrimental in the long term for neurogenesis. And this is really consistent with what we observed when we have the chronic blockade, that again, we have a reduction in neurogenesis. So it seems that this, and this is sort of represented here, that if the early conditioned media from phagocytic microglia is inhibitory for neurogenesis, but this inhibition somehow is important for the maintenance in the long term of neurogenesis. So this is what we think this is important for the homeostasis of the system. So as I was saying, there's a lot more data already published. I will be happy to take questions, but I went kind of quickly in this section because it's done. So, but at this stage we wonder, so is this phagocytosis, does it only affect the homeostasis of surrounding neurons, or does it also affect microglia per se? So to study this project is being done because this is unpublished data by my Marcus in the PhD studies in the lab. So I'm gonna go back to the gene arrays we did in the phagocytic microglia in vitro. And whereas before I focused here on the neurogenesis and in neuron projection, you can see that actually we had many genes that were regulated, that were involved in metabolism. And if we actually draw the genes that are in the glycolysis, pentose phosphate pathway, lactic fermentation, Krebs cycle, and so on, you can see that most of these genes, particularly in the Krebs cycle, are then regulated. And so these were really reminded us of this. I don't know if you are familiar with this concept, the concept of trained immunity. But for immunologists, this is not a novel concept, but is really interesting these days. So what they know is that in macrophages, in the innate immune system, upon stimulation, the first stimulation triggers some metabolic reprogramming, epigenetic modifications that then leads to long-term effects and altered responses. So we wonder whether this phagocytosis of apoptotic cells by microglia was triggering some sort of similar trained immunity. So we started by analyzing the metabolism, and what Mark has found is that we don't really see the type of remodeling that it was observed in trained immunity, where they see is an increase in glycolysis at the expense of oxidative phosphorylation. We actually see a reduction in both glycolysis and oxidative phosphorylation. So it's like the whole microglial metabolism is slowing down after phagocytosis. And some of these, particularly in the oxidative phosphorylation, is really related to a reduction in the mitochondrial network. So we've done some experiments using transfected BB2 microglia cells with a marker for mitochondria, and Mark has done a lot of analysis in counting the objects and analyzing the branching. And what she can see is really not only a reduction in the total amount of mitochondrial mass, but also a reduced complexity. They have fewer branches and fewer junctions. We have also observed a very similar thing in primary cultures of microglia. 24 hours after phagocytosis, using electron microscopy, you can see that in naive microglia, we have many mitochondrial objects, let's say. So if you have a 3D mitochondrial network and you cut it in very thin sections, what you will see is very small, many and very small objects. Whereas in the phagocytic microglia, we tend to have only the larger objects. We don't have the many fewer. So this is really, again, consistent with a less ramified mitochondrial network. So to sort of try to understand why is this metabolic remodeling of microglia going on, or what impact would it have on the microglial function, we've turned our attention to a model that we call a model of super-phagocytosis. So what we do is we use a very low level of cranial irradiation to graze. For instance, if you were studying glioblastoma and you were to give your mice an irradiation similar to what persons would have in vivo in humans, that would be around 8 grays. So this really is a low dose. So this low dose causes apoptosis exclusively here, mostly in the subgranular axon, because it's really targeting the proliferating neuroprogenitors. And what you see here is that everything is cleared off the tissue by 24 hours. So if we go now by numbers, you can see this huge rise in apoptosis. This coupling between phagocytosis and apoptosis is slightly low, so not all apoptotic cells are phagocytosed, but soon, because by 24 hours, everything is cleared out. And we also had this interesting, sorry, I forgot to put the statistics here, but we have this interesting data that I will discuss later on, where we had a trend to decrease microglia and then pump up and then go back to sort of normal levels. So how do they cope with this increase in apoptosis? Is because they have a huge increase in their phagocytic capacity. So this is an example here. You can see to really discriminate between microglia eating apoptotic cells and microglial nuclei, we use PUU.1. And as you can see here, for instance, this cell here is bringing down this process. So this is a cell in the molecular layer that is extending its process and phagocytosing this little apoptotic cell here. And this friend here we have is this one. You can see this is the microglial nuclei, and here is one apoptotic cell. Maybe this is another one broken into 3 pieces, another one, and so forth. So if we calculate the phagocytic capacity of microglia, what's the percentage of microglia engaged in phagocytosis? You can see that by 6 hours, most of them are phagocytic, and they have 1, 2, and even 5 pouches. So they really become super phagocytic, all of them at the same time. So it's very interesting because they are, what's this word? Synchronized in phagocytosis. But then by 24 hours, everything is pretty much go back to normal. So we are doing a lot of analysis to really understand how this affects microglia. I'm going to show you only some of the data that we have. One of the important points is to really assess whether microglia is damaged in their DNA or not. So we use histone gamma histone 2AX, which labels these double-stranded breaks. And I hope you can see there's actually very little colocalization between the red dots and microglia. So you see some damage here in the granule cell layer, but most of the really damaged cells are the apoptotic cells in the subgranular zone. So where we have calculated the percentage of the DAPI area with double-stranded breaks, and you can see there's a rise at 6 hours and then it goes down. And in microglia, please note that this is 1 order of magnitude below the rest of the nuclei in the granule cell layer. This is an example of a microglia with a little bit of these double strand breaks. So microglia is very little damaged, but what's the impact of all of these? What happens to them when they eat, and why we had this decrease in the number of microglia? So we are starting to think that this is very stressful for microglia. So they are very good eaters, but to some extent, this is stressful for them. Because if we analyze Ki67 as a marker of proliferation, you can see we have a lot of cells proliferating here, the regular neurogenic cascade at 6 hours. It fades away between 6 and 24 hours, and then it slowly goes back up. But we have some cases of proliferating microglia. So this is proliferation for all the cells. It really drops down and then it recovers a little bit. So we are not really completely erasing the neurogenic cascade, it sort of comes back. But you can see this trend in increase in microglial proliferation that really matches this increase in the number of cells that we see. So this is one of the reasons, if you put all this together, the decrease in metabolism, the decrease in microglia, the proliferation in microglia. So we have started thinking that this really is, so they cope with this phagocytosis, but to some extent they are stressed and they need to be replaced. And this is why proliferation is turned on. So we are still analyzing this data, to be honest with you, but I think it really brings up new concepts about how we understand microglial proliferation in the context of neurodegenerative diseases. We tend to think that microglial proliferation is always a bad response of microglia because it's correlated with inflammation and with damage and so on. But maybe it's microglia trying to recover themselves for the accumulated damage, whether it's resulting from phagocytosis or not. So we are still exploring this model. I think we have a lot more to learn. But this death of microglia, we have also observed it in vitro. So if we go back to our original model that I showed you before, you can see that over the hours, we have a tendency to have fewer microglia and more pyknotic microglia. So it's not something that is really specific for the irradiation. We think it's really something related to the phagocytosis. And in the gene arrays, actually, this is a horrible figure, but these are all the gene functions, the gene ontology for all the genes that we found upregulated and downregulated in the phagocytic microglia. And I just want you to focus here that we really did observe many genes involved in cell cycle and in death induced by phagocytosis in microglia. So another thing that we learned from this model of irradiation, and it also is very similar to things we had observed before in excitotoxicity and inflammation models, is that microglia really has these three strategies to cope with increased neurodegeneration. They really are amazing eaters, regardless of whether they eat later on or not, but they really can eat a lot. So if normally in the adult hippocampal neurogenic cascade, we have only a few microglia that are engaged in phagocytosis, they have the strategy to recruit more phagocytic cells, almost to 100%, like we see in irradiation. They can increase the phagocytic capacity per cell with up to 5 pouches per cell. And in chronic models, like in chronic inflammation, we also have an increase in microglia. So all combined, this really turns to be a huge reservoir for phagocytosis. But interestingly enough, this is not used when we have some neurodegenerative diseases. We published a few years ago that this process was impaired in epilepsy, and I will show you more data now that we have in a model of stroke. So I think really this brings up the idea that we may need to figure out ways to manipulate microglial phagocytosis as future therapies to accelerate the healing of the diseased brain. So this is again published. I'm going to go back a few years and show you our data with the kainic acid model. So this is a model of epilepsy. So we used intrahippocampal kainic acid. This is a very well established model of temporal lobe epilepsy. And again, you can see this huge increase in apoptosis in the subgranular zone. But unlike in irradiation, microglia are not eating enough eating up enough in this model. And now we don't maintain the phagocytic index around 90%, but it really drops to even down to 10% by 24 hours. So, sorry. So you see we have the increase in apoptosis, but now we don't have the expected increase in the phagocytic capacity. We actually have a little bit of a decrease, and this is unrelated to alterations in microglia. So we actually found out several mechanisms that could explain why microglia become such lazy eaters just when you need them the most, like in this mesotemporal lobe epilepsy model. And one of the things we observed is that many apoptotic cells are really close to microglia. Like this is, for instance, like half a pouch, but it's not complete. So we don't know if this will end up in efficient phagocytosis. And we also have things like, this is like an apoptotic cell in the elbow of a microglial branch. So we measured the distance between apoptotic cells and microglia, and we found that in control conditions, most apoptotic cells are really close to microglia, like around 1 point something micron. But now in kainic acid, we have a bimodal distribution. So on one hand, we had many apoptotic cells really close to microglia, like the ones I'm showing you here. And this really suggested we had a defect in the recognition. So we actually determined that there was the seizures in this model really reduced the expression of many phagocytosis receptors in microglia, like TREM2 and some others. But we also had many apoptotic cells that were really far from microglia, almost at 20 microns, which is like 10 times farther than they used to be. And this really suggested that microglia had trouble understanding where the apoptotic cells were. And this defect in the targeting was related to reduced microglial motility in the hippocampus. To do this, actually, Jorge Valero, a senior postdoc in the lab, developed an automatic filament tracer. It's called PromoID, and this is automatic 3D analysis of microglial motility. I really recommend you to go ahead and check it. This defect in phagocytosis was not only occurring in the early phases, but it was also in the long term and also in humans. So these are examples of 4 months after seizures induced by kainic acid. We have here an apoptotic cell sitting by this reactive astrocyte, not being engulfed. This is an example of phagocytosis. And we were able to obtain human MTLE tissue from biopsies from patients that, so unfortunately, many of these patients are pharmacoresistant, and then they have to go through surgery to remove the focus, which in many cases in the hippocampus. So it's very unfortunate for the patients, but it was lucky for us because we could really obtain this tissue within 30 minutes. So we didn't have any postmortem delay, and we could really assess phagocytosis in the human brain, which for the most part is very similar than in the mice. Using this, we call it ball and chain, is like in terminal process. So microglia extends the process and grabs the apoptotic cell. But we also had some cases of an apoptosis, an apoptotic cell that was being phagocytosed by like a master-like structure. I call it the noodle soup, where you have many microglia, like this one and this one and this one, all of them throwing their process just to make sure that this cell is sitting up. And this is really a messy thing. So phagocytosis was still reduced 4 months after kainic acid in the mice. And humans, even though we do not have controls, because again, this is biopsy tissue, they don't have much levels of apoptosis. Only 50% or under 50% of the apoptotic cells are engulfed, which is not a very good measure anyway. We had another study with control and epileptic brains in the proper controls, I mean, in autopsy tissue, but autopsy tissue is not very good for phagocytosis because of the postmortem delay. So this is not specific, this impairment is not specific for epilepsy. We also find it in stroke. And this is work by Solveig Kari, again, a PhD student in the lab. And what she has been using is a model in which she introduces a filament through the internal carotid artery. She can do the occlusion and then the reperfusion. And she sees something very, very similar along the time course. Again, she sees an increase in apoptosis, a damping in the phagocytic index, and microglia not being able to respond whatsoever to these apoptotic cells. It's like they don't see them, they don't notice them. And as a result, the coupling between apoptosis and phagocytosis is lost. So this is very bad for the tissue. The mechanisms are to some extent similar, to some extent different. So one of the things that we know, and this is work by Noah Zavala, a postdoc in the lab, is that if we use organotypic slices and we do reperfusion, So microglia is quickly blocked by the absence of oxygen and nutrients, but these can really turn back really fast if there is proper reperfusion, which we don't have in the MCAO, in the medial cerebral artery occlusion in the model I just showed you. And this is also related to a decrease in motility. You can see here by the distribution in the process velocity that microglia are really less motile once they don't have oxygen and nutrients, as you would expect. We also have experiments showing that this is not only affecting the motility and the engulfment, but also affecting the degradation, which really suggests that there is a defect in the lysosomes. And this is work by Virginia Sierra Torre doing her PhD in the lab. She has used primary cultures of microglia that she feeds with apoptotic cells. And now we feed them and then we remove them. It's like a single exposure to apoptotic cells. So now we can really trace what happens to them, and we can discriminate between the efficiency of engulfment and the efficiency of degradation. And she has observed this defect in apoptotic cell degradation that suggested that the lysosomes were not working properly. And we have some preliminary evidence that they do increase their lysosomal pH. Again, probably because of the lack of oxygen and nutrients, that it's somehow disturbing these proton pumps in the lysosomes. So altogether, really suggested that this impairment of phagocytosis is important in more than just epilepsy, also in stroke and maybe other diseases, and really turned out are interesting to trying to figure out ways to manipulating it that could be used in the clinic. So Iñaki País, he has appeared before again for his PhD thesis in collaboration with Jorge Valero and nearby company called Innoprot. We used our in vitro model to again discriminate between engulfment and degradation, and we used compounds from the Presswick library to try and find these drugs. So, sorry, this picture is wrong. Just don't look at it. But what we had is a list of candidate drugs that then we organized degradation pro and anti and engulfment pro and anti, and we are currently testing them in organotypic cultures and soon in vivo models to see if we can really manipulate phagocytosis in vivo and see if it really can help to accelerate the healing of the diseased brain. So I think that was all. I'm just going to finish by thanking all the members in my lab. Everybody worked really hard to get all this data. Of course, our collaborators and our funding agencies, and all of you for your attention. And I will be very happy to take your questions.